How are you? My name is Michelle Connor. I'm your VCS for Living Environment. Today we're going to be looking at the herd immunity simulation. This is a really cool simulation. It's very simple, and once you know how to manipulate everything, it tells you a lot of really good information. So again, for the teacher view on the first page is really where you can really grab everything that you need. There's an Understanding Herd Immunity Lab, which is like a direction sheet, the actual simulation, the master data table, which you're going to assign to every student, and then pulling class data, you're going to need one copy for each class. So just to go over, you have the simulation looks like this. The directions are here. This is the one that's going to get for each student's going to get. And here's where we're going to pull the information. So the simulation is actually very simple. So if we just look at the directions really quick, what you're going to be doing is you're going to be going in and you're going to kind of be manipulating some of the data. So we're really only going to be changing the immunization rate. So you'll notice the immunization rate on the left and on the right, we're going to make sure that it's the same, which is automatic. The 0.9 is the default value. As a teacher, you're going to go ahead and give students different numbers as far as the immunization rate. In order to see like the difference and like when herd immunity is reached. So the values are going to range from 0.1 to 0.9. Now what happens is as you change that, the students are going to go ahead and actually infect certain unvaccinated individuals in the simulation, which are the red symbols, and then they're going to get some results that they're going to calculate. So it sounds a little complicated, it's really not. So if we look at the simulation, it's very simple. The green is vaccinated, the red is unvaccinated, and then anything that's an X is going to be infected. So if we look at the bottom, here's where you're going to get your information. We're going to change this to 0.6, let's say. Now you'll notice, since this is clicked, it's the same. As the immunization rate drops, the more red unvaccinated cells you'll see. Then your students are going to go ahead and infect one of the unvaccinated cells. So all they do is they click one. Now, as they click, you'll notice anything that's an X is now an infected cell. This is not good. Now, obviously, some of these green circles, which are vaccinated, are also getting sick. So with one person getting infected, the results are here. So it says 52.9% is infected, 34% of vaccinated is infected, 96% of unvaccinated people get infected, and 34.3% of infected that had been vaccinated. So here is where they're going to take these numbers and put them into their master. So population infected. So the rate would be here 0.6. Population infected. They'd go ahead and drop these numbers in, and then they're going to be able to get an average. So we're going to do this for 3 trials, and they can click anywhere. So what we're going to need to do is we're going to need to repopulate. And then we'll go ahead and we'll pick a different one. And we'll get our numbers. Once they get their numbers, you'll notice this is still going. They're going to take their percentages again, put it into their data table, and then once they get their averages, they're going to go ahead and they're going to write their names, their assigned immunization rate, and then their average numbers, and they're going to be able to collect the data as a class.